Hello, dear students. Myself, Dr. S.K. Prajapati. Today, I am going to deliver my lecture on monoclonal antibody. As you know, antibody is very important aspects of our discussion today, because today we will discuss about their principle, their procedure, their type, their limitation. So, what is monoclonal antibody? As the name is indicating, we will discuss further. Monoclonal antibody are defined as the antibody derived from a single clone of plasma cells. Plasma cells, where it is available inside our body, and in our body there are various types of cells are available, and particularly beta cell, B cell are responsible for defense mechanism. So, all having the same antigen specificity, that is produced against a single epitope of an antigen. Now, we will discuss further polyclonal as well as monoclonal antibody. So, we will discuss on the following topic. Number 1 is principle. Principle means what is the basic principle, what is the scientific reasoning behind the production of the monoclonal antibody, procedure, how it is implemented, how we can perform the overall procedure for the production of monoclonal antibody. After the production, after the processing, we have to purify it, we have to culture it by using various medias. Then after that, what will be the fate of the selected cells which we are using for the monoclonal antibody. Type of monoclonal antibodies is the another step, and after all, finally we will discuss the application of monoclonal antibodies. So, on these parameters we will discuss one by one. When an antigen having multiple epitopes, as you know, as you have learned in the basic immunology, that how the antigen and antibody are produced. So, I am not discussing that one, just we are coming to that an antigen having multiple epitopes enters the body. Each epitopes may stimulate one clone of B cells. Means B cells, it is available in our plasma, and it is responsible for producing the special type of antibody against that particular type of epitopes. If it is having one epitopes, it can produce one antibody. It is having two or more, then it can produce multiple antibodies. So, overall responding cells are B cells. Now, resultant antibody mixture present in serum is polyclonal in nature, because if it is having one type of antibody, it is called monoclonal, because we are just going to start perform the processing. So, here the various type of epitopes which is available at the site of the antigens, and as per that our B cells responding. So, in that context it is called polyclonal in nature, that is contains mixture of antibodies derived from different clones of beta cell. Next is, because one organism contains many different epitopes, the host serum contains various polyclonal antibody. So, this is basics, that how much epitopes, how much sites it is having for infection, as per that our body will respond. Body means our B cells will respond. So, polyclonal antibody used in immunodiagnosis are prepared by immunizing animals. Now, how we are going to produce these antibody, it is very important. So, by using the rabbits, sheep, and goats. So, with an infection agent, particular type of infection agent we are going to use, and then isolate and purify the resultant antibodies from the animal serum. Are you getting it? Means which type of antibody you are going to produce? You have to isolate that particular antigen, and that particular antigen we can say infectious agent. It may be anything. It may be anything which are responding for any type of infection inside our body. So, that particular antigen we will inject in the animals, and after the isolation of their plasma, what type of cell it will produce, we will isolate, purify, and further we will understand one by one. Now, what is the principle behind this? It is very simple principle of the production of the polyclonal, or we can say the monoclonal antibodies, because it contains the mixture, then it is polyclonal. So, a clone of B cell stimulate against a single epitope antigen, that is antigen producing plasma B cell, is fused with malignant antibody producing myeloma cell to produce a hybridoma cell. So, this is the basic of this principle, that one we need B cell and one we need myeloma cell. These two are very important. So, again I am repeating that a clone of B cells stimulate against a single epitope antigen, that is antigen producing plasma B cell, is fused with malignant antibody producing myeloma cell to produce a hybridoma cell. Hybridoma cell we can say hybrid. It is a hybrid of single beta cell, B cell and myeloma cell. Okay. You should not confuse with the beta cell, because it is available inside the Langerhans, isolate of Langerhans. So, I am telling you these B cells are B lymphocyte cells, we can say the. Okay. So, here one cell B cell, one myeloma cell, these two cells are important. After fusing it, they are called hybridoma, hybrid cell. This hybridoma cell has two unique properties. It is very interesting. Now, one step is interesting, then one healthy cell B cell, one myeloma cell. Myeloma cells, you know, cancerous cell, because why we are going to choose the cancerous cell, I will discuss later. So, it possesses two unique property. This is the whole concept on which this particular principle is standing on. Number one, produces monoclonal antibody of same antigen specificity due to B cell component. B cell, if it is going to develop in the future, then it will produce their progeny in the same way as the property possessed by the B cell. Another one is multiply indefinitely, producing a clone of identical cells due to immortal myeloma cell component. Multiplies indefinitely. What happened? B cell having their own life, as you know, our blood cells having their own limitation, their life cycle. So, they may be survived for 10 days, 15 days, 1 month, 3 months, whatever it may be. But after sometimes there is the greater challenge of survival. Okay. So, now another property which we have to introduce in this monoclonal antibody, that is immortal property, and this immortal property we can obtain from the myeloma cell, So, myeloma cells, and another one is the what we can say the B cells plus myeloma cell. So, these two property is very important in this case. Now, after understanding the principle that one B cell, one myeloma cell, and we are going to develop a hybridoma, and this possess after the we can say after the fusing, it is going to develop which type of property. So, it possesses two specific properties. One is antibody B cell property, possess B cell property. Another one is immortal property. Why it is necessary the immortal property, we will discuss further. So, the process start by immunizing a mouse with the antigen for which an antibody is to be produced. This is the first step. So, the interest means antigen of interest we have to inject in the mouse. It is called the immunizing procedure. So, production of monoclonal antibody, first of all mouse splenic B cell, mouse after injecting the antigenic injection in their peritoneal cavity, we will isolate their serum. So, from where we will obtain. So, here is the step is mouse splenic B cell we will isolate. The mouse is injected with an antigen containing the desired epitopes. Epitopes, why epitopes? Because it is going to potentiate the antibody, going to produce the antibiotic response. So, the animals responded by producing many antibodies, the two epitopes injected. Okay. So, against that epitopes, various type of antibodies are going to be developed. Okay. After an interval, the mouse spleens which contains antibody producing plasma cells is removed and emulsified, so that antibodies producing cells can be separated out and placed into individual bells. Bells means we can say the particular trace available which contains the microvells available in the laboratory, and after the processing, how will you perform it after injecting it, and then you will leave it for the some time for their survival, and after when it will grow in quantity, and after that you will remove the particular organ, isolate the antibodies, and finally you have to place it in the wells. Wells we can say the simple, it is a tray like a structure, and we can say the generally used in the laboratory. So, these cells cannot remain viable in the cell culture for long time, means which cell? Beta cell, point to be noted here. This is very important. So, these cell cannot remain viable in the cell culture for a long time, so they must be fused. Now, we have injected any type of we can say antigens, leaving it for some time, isolating the organic, and finally we can say the plasma we are going to obtain, and that plasma we have to separate it out in such a way, leave it for the culture, and here again the final step is we are going to discuss, that is having the limitation of their life. So, now we have to fuse with cells that are capable of surviving and multiplying in tissue culture. As I had discussed in the previous slide, that one B cell, one myeloma cell. So, now there is a need of the myeloma cells. Such type of property, surviving, continuously surviving property, immortal property are possessed by the myeloma cell. Now, role of myeloma cell is very important. Myeloma cells are used as a source of such immortal cells. They are cancerous plasma cell. They closely resemble mouse B cells, hence are compatible for fusion. Myeloma cells from where we are going to get. Okay. So, cancerous plasma cells, why we are going to choose this type of cell? Because it is resembles in nature with the mouse B cell. Okay. So, however, myeloma cells also have the capacity to produce their own antibodies. Now, here is another problem, because if myeloma cell is there, then it will also produce their antibody. Okay. Hence, myeloma cells are genetically modified with two mutations, double mutation myeloma cells, so that they lose the ability to produce their own antibody what retain immortal property. Now, point to be noted here is our principle that one cell with myeloma cells, but one antigen is producing antibody, another cells which are having the immortal property myeloma cell is also going to produce the antibody. Now, this may be the problematic. So, after doing some genetic changes in that myeloma cell, we have to just isolate their immortal property. We have to stop their antibody production. Are you getting it? Means now the immortal cell are going to produce their antibody. So, in our body we have to stop it. After how we can stop it? After doing some genetically changed to 2 times, 3 times, whatever it may be. So, just we need immortal property. So, we need particular antibody bind or fuse with immortal property of the myeloma cell. This we need, okay, because we want to continuously grow it. So, for that purpose, for healthy antibody, we need a special antibody should be fused with this myeloma property, or we can say the immortal property. Now, these cells, one important property it possesses at the biochemical level, these cells are also deficient in enzyme hypoxanthine guanine phosphoribosyl transferase HGPRT. Simply we can say the HGPRT enzyme. This HGPRT enzyme play very important role. Further, because our condition is just we need immortal property, we do not want to produce antibody from the myeloma cell. So, we are going at the biochemical level, what is the role of the various enzymes. Okay. So, these cells are also deficient in the enzyme hypoxanthine guanine phosphoribosyl transferase HGPRT. We can say this defect lead to their inability to survive in a medium contain, or we can say the head medium, and we can say containing hypoxanthine, aminopterin and thymidine. So, this is called head medium. This play very controlling role, we can say. What? HGPRT controlling role. So, this is the defect. This is the defect of the cells. The defect leads to their inability to survive in a medium containing hypoxanthine, aminopterin and thymidine. Okay. It is called het medium. Next comes to the another step, that is fusion. Now, we are going to fuse. What we are going to fuse? One beta cell, one myeloma cell. There is not only one, but we are going to discuss in the discussion one beta cell, one myeloma cell. So, fusion the mouse splenic B cell and mutate it, because we do not want the antibody response of myeloma cell, just we want their immortal nature. Okay. Get it? So, here we are going to fuse. So, that mutated myeloma cell which loses the antibody production potential, just it is having immortal property. So, here we are going to fuse in presence of polyethylene glycol. Okay. So, in this reaction chamber, we can say in this reaction chamber there are 3 type of cells are going to produce. Number 1, unfused myeloma cell or cancerous cell. Another one is unfused mouse splenic B cell, and third one is fused hybridoma cell. You get it? It is important step. One cell, that is unfused myeloma cell, one cell, that is unfused mouse splenic V cell, and fused hybridoma cell. Means which cells are going to fuse with the each other, it is making the hybridoma. So, it is called hybridoma cell. Now, what events? Purification after the fusion, we are going to purify the cells. So, first of all we have to culture it in the head media, hypoxanthine, aminopterin, thymidine. So, this media contains these three components. The next step is to remove the unwanted unfused cells and to propagate the clone of hybridoma cell, because we have already stepped up, we want to remove unfused myeloma cells, we want to remove that unfused beta cell. Just we are having the interest about the fused hybridoma cell, and these fused hybridoma cells just we have to subculture. So, we have to propagate it. This can be carried out by subculturing the cell in reaction chamber into a special medium, that is called head medium. Okay. So, here is again is an interesting topic, because we had already discussed the cells should be genetically modified, and another one is it should not possess the antibody production property of that myeloma cell. It should be possessed by the beta cell, B cell. So, further what is other important aspects which we have to give attention, that is mammalian cell splenic V cells synthesize purine by either de novo or salvage pathway. As I had discussed, that SGPRT enzyme is responsible which are deficient in the myeloma cell. Here our B cell, our means mammalian B cells are used to pathway for their basic material which we have to use for the DNA synthesis. Okay. So, here aminopterin blocks the de novo pathway, aminopterin blocks the de novo pathway, so that the cell has to perform the salvage pathway to synthesize purines for its survival. Okay. Means salvage pathway it will use, de novo close, salvage it will use. Two pathway are there, one due to aminopterin in head media, so it blocking, it is just opening the salvage pathway. So, salvage pathway requires two important enzymes. This salvage pathway requires two important enzymes, one is SGPRT and another one is thymidine kinase. You get it? Okay. So, myeloma cell which are deficient in SGPRT, get it? Okay. So, a myeloma cell lacks SGPRT, it cannot grow on head medium, but antibody producing spleen cell can survive as they possess this enzyme. Okay. B cell possess, but myeloma cells are lack in SGPRT, so it can't follow this de novo pathway. Okay. So, it can't survive. Now, hybridoma cell, fused hybridoma cells survive in the selective media and can be recognized by their ability to grow indefinitely in the medium. Only hybridoma cell possess this property, because especially myeloma cell are lack in SGPRT. Okay. So, unfused splenic cell and unfused myeloma cell. Unfused myeloma cell they cannot grow as they lack SGPRT enzyme. SGPRT it is not there, so it can't grow further, means it can't use that particular pathway for their purine synthesis. Unfused splenic V cells they can grow, but do not survive for longer a period of time, because it is not having the immortal property. Okay, so if the original antigen used has multiple epitopes, many B cells would fuse with myeloma cell to produce a mixture of hybridoma cells, each having a specificity of one epitopes. The medium containing hybridoma cells is then diluted into multi-vase plates so that such extent each well contains only one cells. After this, we are going to purify this antibodies by using the microwell techniques. So, we have to isolate a specific type of antibody and we have to culture it. We have to culture it, either we have to culture it, either we can isolate some specific type of antibody of therapeutic interest, and we can process further. So, various type of tests are there for their separation on the basis of their qualitative as well as quantitative aspects. So, ELISA technique we can use for this purpose. When a good candidate antibody producing cell is found, the hybridoma cell are selectively proliferation either in cell culture or in vitro, or they can re-inject it. There are two ways. Either we can suppose after isolation we can culture it again and again for increasing their quantity, or we can again re-inject in the mouse and again we repeat the process for their multiplication. So, where the cells multiplied produce in large quantity, and again we can use various type of techniques for their isolation. We can use the chromatography, we can use the various immunoprecipitation test for their isolation, or we can say the purification. Now, we are coming towards the types of monoclonal antibody. This is we have to clarify it, because one antibody we can produce with the help of animals. Once when we produce the antibody from the animals, if we are going to inject in the human being, it may be treated as a foreign particles. So, may or may not it can accepted by our body. Okay. So, on that basis particular type of antibodies are very important. Now, we are we can say that the above mentioned procedure would yield monoclonal antibody whose 100% amino acids are mouse derived. This is the basic problem, because we are just separating out from the mouse, and whatever their amino acids, we can say the genetic information which are not acceptable in the human being, and it is treated as a foreign protein. So, can induce immune response in human, producing human anti-mouse antibodies. Now, this is another problem. If we are injecting that one antibody in the human being, then it can produce HAMA, HAMA means human anti-mouse antibodies, antibody producing against the antibody. So, hence mouse derived monoclonal antibody are not the best candidate for the human use. Now, this is the problem. So, further, since the discovery of hybridoma techniques, various modifications have been attempted by the various scientists or researchers producing the monoclonal antibody by recombining human and mouse protein. So, by applying the genetic engineering, as you are familiar with this terminology also, we can classify the whole monoclonal antibody. Now, this slide is showing that is mouse monoclonal antibody, chimeric monoclonal antibody, humanized monoclonal antibody, or human monoclonal antibody. So, by showing this slide, you can understand there are 4 type of antibodies are shown, that is mouse antibody. What is mouse antibodies? Here the term which is indicating OMAP, it is very important because it is used in the classification of the monoclonal antibody. So, it contains 100% mouse derived proteins. The property of this mouse antibody, it is we can say the 100% mouse protein. Okay. They can lead to an allergic reaction in human. This is the limitation of this mouse monoclonal antibody. Another one is a chimeric monoclonal antibody. It is represented as XI-mab, as you can see in the slide. So, it is prepared by recombine of mouse protein plus human protein. Human protein, these can also cause an allergy. Another one is humanized monoclonal antibody. It is represented as a GU-map. Here it contains the mouse as well as the human proteins also. So, the ratio of mouse proteins is very less, humanized ratio is very we can say that is up to now 90%. So, it is represented by the GU-map. Another one is human monoclonal antibody. It is 100% human derived proteins or amino acids, and it is represented as a U-map. So, this slide is showing the structure which you can go through. Please give their attention that the Y-shaped structure having 2 light chain, 2 heavy chain, and these 2 light chain and 2 heavy chains are the representative of that particular antibody structure. In that, one is heavy chain, one is light chain. So, in this figure totally mouse antibody are there, chimeric antibody are there, humanized and human antibody are there. So, by understanding by seeing this picture, you can understand in a better way that how the type of antibody we can understand. Next one is, this slide is showing the examples of few antibodies of chimeric, which is represented as XI. It is the example is FC-map, G2C-map, and humanized type of antibodies represented as ZU, Pali-V-ZU-map and Veva-C-ZU-map. This is the example of the antibody of humanized antibody. And another one is human. It is represented as a U. It is Ipli-mu-map. So, this is the example. In that nomenclature, generally this terminology is used. So, on that basis we can understand which one is chimeric type, which one is humanized type, which one is human type. So, finally, we are coming towards the applications of the monoclonal antibody, and this monoclonal antibodies isolation and purification of various type of substances, we can say, especially interferon and coagulation factor 8. It is used for their isolation and purification. Another identification of cells and clones we generally use, especially TH and TC cells, by using the anti-CD4 and anti-CD8 monoclonal antibody. Diagnostic reagent, generally the various type of kits are available. In that the various monoclonal antibodies mixtures are used. On the basis of the fluorescent we can detect any type of diseases. Okay. Next is detection of infections. Various type of antigenic infections we can detect with the help of the antibody, that is hepatitis B and infection of streptococci. As you know, most popularly example is pregnancy detection test is there, and by using the monoclonal antibody against human gonadotropin hormones. So, nowadays you have seen various type of kits are available, that is work on just putting the samples, and you can identify which type of disease are there. Blood grouping we can detect, tumor detection generally we identify with the help of the various type of antibodies mixture, especially in the case prostate specific antigens. Tissue typing anti-HLA monoclonal antibodies we use for the transplantation of the tissue, monitoring of various type of proteins in the and the drug in this serum. So, nowadays there are very we can see the broad use of the antibodies, and just we can say some of the therapeutic uses also we can discuss here. Monoclonal antibodies are used in the treatment of various inflammatory and allergic diseases and cancers. So, useful for treatment of the various type of cancers, and nowadays various type of kits are available. The most important step is that is US FDA has approved more than a dozen monoclonal antibodies, that is alemtuzumab and trastuzumab, to treat certain cancers, and the mechanism by which the monoclonal antibody works as a therapeutic agents are suppress the immune system, kill or inhibit malignant cells, inhibit by using the angiogenesis. So, thank you students. I hope that you understand and cleared your doubts. Thank you.